Okay, sorry.
Hashtag drunk.
So eukaryotic cells are larger because they have more stuff in them. They have organelles. They have a nucleus. Prokaryotic cells, no nucleus, extremely small unicellular organisms. So, yeah. Eukaryotic means true kernel. I guess nucleus is kind of like a kernel. And prokaryotic means that it doesn't have a kernel or nuke. I always tell my kids, nuke, pro, no. Even though pro really means like you're for it, but it rhymes with no. Okay? So, nuke, that's the safe one. All right, discovery of cells did not occur until we had the microscope. And so Robert Hooke is the first guy to coin the term cells. He was looking at dead cork cells, and you notice all these open spaces, and he's like, oh, they look like tiny little jail cells, so I'm going to call them cells. Anton von Leeuwenhoek looked at pond water and noticed that there were living microscopic organisms swimming around in the and everyone started to look at everything underneath the microscope. It took 150 years for the cell theory to develop, and it was put together by three guys. Matthias Schleiden, 1838, noticed that all plants were made of cells. He looked at different types of plant tissues, and he found the same result: cells, 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 cells. Theodor Schwann, a year later, started to look at different animal tissues, and he also noticed that they were made up of cells. And then they don't mention Virchow, poor Virchow. Virchow was the first guy to see cells split underneath a microscope, actually divide. So when he, stated what he saw, everyone thought it was kind of crazy, and then other people started to confirm his observation, and so he's responsible for the third statement that states that cells arise only by division of a previously existing cells. So 3 principles for the cell theory. All organisms are composed of one or more cells, and the life processes of metabolism and heredity occur within these cells. Second statement, cells are the smallest living things, the basic unit of organization of all organisms, and then 3, cells arise only by division of a previously existing cell. So I always say cells are the basic unit of life. Cells make up living things. Cells come from other cells. However, if you remember from our chapter 2, I believe, we talked about the chemical hypothesis. So life had to evolve spontaneously at some point to go from non-living to living. So, but in today's world, we haven't come across a living thing that hasn't been made of substance.
What if we do?
Then we got to change the cell theory. We throw out the first 2 statements. All right, so cell size is limited. They are small due to diffusion rates. So how fast they can move things affects their surface area. The temperature also affects how fast they can move things, as well as concentration gradient. So in science, whenever you see brackets in a word or a chemical symbol, that means concentration. Okay? So as a cell size increases, your length of diffusion also increases. And so those larger cells need to make more macromolecules. They need more energy. They also have to get rid of their waste products. So if it's too big, that means they can't move these things fast enough, and then the cell suffers. And so cells have an optimal size of being small for that increased surface area volume ratio. So as the cell size increases, volume increases more rapidly than surface area. And so you're looking for the ratio. 6 divided by 1 is 6. If you were just to double the size of the cell, now your volume, surface area is 24, volume is 8, so then it's a ratio of 3. So this is highly more optimal than this one. And if you jump to 4, well, you can do the math, but it's not a very good ratio. Okay, switching gears, talking about microscopes a little bit. So without microscopes, we wouldn't be able to see cells. However, microscopes do have a problem, and it's called the resolution problem. We can only see as well as how good the resolution of a microscope is. And so the definition of resolution is the minimum distance between 2 points that can be apart and still be distinguished as those 2 points. And so a lot of times you'll see things that are blurry, but then as they start to separate and you can see the 2 points, well then now you have some clear resolution. And so here it's just a little scale to show you, an ant's about 1 millimeter, hair is 100 micrometers, a cell between this, it's got to be eukaryotic. Bacteria are a lot smaller, between 1 to 10 micrometers, but then for viruses and proteins and small molecules, well, you can see viruses, you can see some proteins, but we haven't really been able to see There are different types of microscopes out there. So one way to increase resolution is to increase the magnification to help with that problem. So we have light microscopes where you use visible light, and there's 2 magnifying lenses. We have compound microscopes that magnify in stages. So you have like an eyepiece and maybe a couple of objective lenses to magnify it. So maybe to solve this resolution problem, you can be like, why not just keep adding another lens to really magnify things? Well, it has something to do with light and physics. So maybe in physical science, you guys did light ray diagrams? No?
Is that where like the concave and the
Yes.
Oh, I remember.
So you had a diagram like this, and the light ray came, and then went through like the focal point. Oh yeah.
That was terrible.
Yeah. So the problem is, however, those light rays are actually gonna start overlapping at different spots, and so you actually have to change your wavelength. You can't have visible light anymore, and so now we use electron microscopes. So electron microscopes, 1,000 times the resolving power than a regular light microscope. There are 2 types of electron microscopes: transmission and scan-in. Transmission goes through the specimen. You get a 2D image, as you can see here. So imagine like taking a cilia and just go, and then just looking at it from above. You can see circles. Well, that's the cilia that get cut, and you're looking through it. So these electrons travel through the specimen, giving you 2D images. Scan-in electron microscopes, the electrons bounce off the object, and you get this really cool 3D image, so you can actually see the cilia kind of flow back and forth. This is a light microscope of a cheek cell.
So the stem is better than the
Depends on what you're looking at. Because if you're looking at things inside the cell, then you want transmission. But if you just want to look at the outside of the cell surface, like how does a virus attach to a cell, well, then you might want to look at scan. So they're both beneficial.
Have you ever like watched?
I have never in person, but I've seen pictures. So, and you'll see some too. We'll actually dedicate like a whole section to viruses, and then we'll talk about possible Eukaryotic disease and prions and a few other things. With light microscopes as well as electron microscopes, you can stain your image to contrast different cellular components. So at Gustavus, they have a really fancy microscope that's like $250,000. And when I took cell biology, I got really good with it. So then I started to do all of the staining and analyzing of cells and taking pictures of cells and giving them to the professor and all the lab assistants. But there are different colors that you use. And we always use a Dappy blue for the nucleus. So what you're seeing here is just the nucleus being stained. Green is usually the cytoskeleton, like the outside kind of tubulin. And then red is also part of the cytoskeleton, but it's a different component. And so then when you overlay all 3 of these, you get a cool image like this. Okay, some basic structural similarities across all cells is that it has genetic material. In prokaryotes, remember, they do not have a nucleus, but in the center of their cell, they have a dense genetic region. It's called the nucleoid. Eukaryotes have a nucleus, and the nucleus is a double membrane structure. And so we're going to actually take a look at the nucleus structure here, I think today, if not tomorrow. So that's one similarity, genetic material, centrally located. The second similarity that all cells have is they have a cytoplasm. It fills the entire interior of the cell. It's this aqueous solution, contains sugars, amino acids, proteins that the cell uses to carry out everyday activities. It's more jello than water because of all this stuff. This is where you will find the organelles, which are specialized membrane-bound components that carry out different functions for the cell. You may have heard the term cytosol. It's very similar to the cytoplasm. The cytosol is any part in the cytoplasm that has ions or molecules dissolved in solution. Okay, so that's what they mean by cytosol. You can kind of use them interchangeably.
Sounds like a cleaning product.
Okay, the third thing that all cells have in common is that they have a plasma membrane. which I will abbreviate PM from now on. So that doesn't mean Prime Minister, it's plasma membrane. So job of the plasma membrane, it closes the cell, separates it, the inside from the outside. It's made up of phospholipid bilayers, okay? And so you get the head with the two hydrophobic tails, and the tails interact with each other, and the heads are polar, so they interact with aqueous solution inside and outside the cell. Inside the phospholipid bilayer, you might see proteins or cholesterol or carbohydrate chains attached to it. And what they're doing is they're just interacting with the environment. They might be responsible for turning on a signal. But we do have transport proteins. In fact, on Friday, you're going to read an article about aquaporins, protein channels that allow water to enter and leave the cell very rapidly. Aquaporins are tied in with kind of like biofreezing, like how frogs, some certain
Your
species.
attention, please. Teachers, please do lunch count for today's lunch and email me. And please do lunch count for tomorrow's lunch and put in JFC as usual. Thank you.
Okay. How some species can lower their metabolism and then go into a hibernation and basically like freeze and then re-thaw and come back out. So we'll talk about aquaporins on Friday. Well, you'll read about it, I should say. Receptor proteins, what they do is they induce changes within the cell when they come in contact with something. So like there are molecules out there that will connect with this carbohydrate chain and start a chain reaction inside the cell saying, hey, this is what you need to do next. They can also function as, that's supposed to be markers that identify the cell. This is how your white blood cells, your immune system works. They go around, they attach to your cells, they read these markers. If the markers are saying that you belong to the body, they leave you alone. If you are foreign material, then they terminate you, okay? Because you don't got the right markers, you don't got the right ID. Okay, fourth thing that all cells have in common is ribosomes. And so the ribosomes, their main job is to make proteins or synthesize them. So this is what a ribosome looks like. It's usually made up of two subunits, a large one and a small one. They latch together. They'll read mRNA, and they make proteins. And so you can find ribosomes on the ER. You can find ribosomes floating around in the cytoplasm as well. Okay, switching gears, let's focus on the prokaryotic cells here. So you can either be a eukaryotic or prokaryotic, so we'll talk about prokaryotics first. Main difference is that they have no nucleus or internal membrane system or organelles. They don't have organelles, they do have ribosomes. I'll talk about how some cells get away with not having organelles, because like there are cyanobacteria, and you're saying, wait, Mrs. Erickson, they don't have organelles. What organelle carries out photosynthesis? The chloroplast. You mean to tell me they don't have chloroplast, but they can still carry out photosynthesis? That should be blowing your minds right now. So we'll talk about how bacteria cells get away with that. So yes, you can be prokaryotic or eukaryotic. So we're going to talk about bacteria or archaea. Then we'll switch gears and talk about eukaryotic. We'll have a whole chapter dedicated to protista, a whole chapter dedicated to fungi, as well as animals and plants. So like, we'll really get in depth with these guys. So prokaryotic cells have a relatively simple organization. They are the simplest organisms. They are usually unicellular. They're small. They have a cytoplasm surrounded by a plasma membrane. They have a cell wall. And so we're going to talk about what that cell wall is made up of. No organelles except for ribosomes. They don't really have a cytoskeleton on the inside, and the reason for that is they have a very rigid cell wall that I will talk about in depth here. The strength of the cell comes from the cell wall made of peptoglycan. There are two domains of prokaryotics, archaea and bacteria. You find bacteria everywhere, or prokaryotes everywhere. They are responsible for a lot of diseases. There are just many different types of prokaryotes out there, and they are responsible for a lot of photosynthesis and the oxygen that we are breathing today. So let's look at the cell wall made up of peptoglycan. It's made of carbohydrate matrix, basically a bunch of sugars cross-linked together, as you can see here, as well as some polypeptide units. So not only is it carbohydrates, but polypeptide, peptide should tell you what type of macromolecule. Protein. Okay, so it's a carb-protein cell wall. Helps maintain shape, prevent excessive uptake and loss of water, because bacteria, if they, their environment does change very rapidly, So they don't want water rushing in and causing it to explode, or water leaving the cell causing it to, well, just dehydrate. Plants, fungi, and most protists have cell walls, but they don't have peptoglycan. So that's the difference between the cell wall of a bacteria and a plant. Plant cell wall is made up of what? Can't digest it.
Oh yeah, it's the
Not chitin.
carrot.
Cellulose. Okay? So cellulose. Fungi is chitin. Protists kind of depends on which protist you're looking at. But bacteria, it's peptoglycan.
Well, is there, cause the peptide bonds are the
Yeah. Yes. Yep. Okay, so there are actually two types of cell walls that you can find in bacteria. And so maybe you've gone to the doctor and they'll swab your throat. Maybe they're checking for strep. And what they're really doing is they're trying to determine what kind of bacteria you have, or if the bacteria is either gram-positive or gram-negative. So maybe you've heard that term before, and you're just like, I don't know what that means. Well, now you're going to find out. So if they say gram-positive, that means that the peptoglycan cell wall is a single layer, it's extremely thick, and it retains a violet dye. So it's purple. That means gram-positive. Okay. Gram-negative is a little bit more complex. These guys are nasty. They're a little bit more difficult to treat. Multi-layer cell wall does not retain a violet dye. So therefore, you might see it as red or pink. So negative, maybe think like, oh, the teacher used a red ink on my test. Ooh, that's just so negative. You know?
Positive purple, and the other one's not.
Well, I'm just saying because this is pink. Pink is also a letter P. So I don't know which way you want to remember that. Okay, so susceptibility to antibiotics depends on the structure of the cell wall. And so I'm going to show you diagrams of these two and then talk about how some antibiotics actually work. Some can secrete a jelly-like capsule on the outside of the cell. These guys are, like I said, the most dangerous ones. They're disease-causing. They'll stick to surfaces that will help support their growth. So here's the difference between gram-positive and gram-negative. Here's gram-positive, one-layer, thick peptoglycan cell wall. Gram-negative, multi-layer. It's got lipopolysaccharides. Lipo, what do you guys think that? Lipids, but then not only that, polysaccharides, sugars, carbs. So this is fat, sugar, outer layer on the inside. Now you get the peptoglycan, which is the protein and carb layer, and then finally you get the phospholipid bilayer, and finally you reach the cell membrane. Now, these guys are nasty. They'll secrete a capsule on the outside. Antibiotics can't drill holes into them because of this lipopolysaccharide. It's too slippery. So that's how antibiotics work. Sometimes antibiotics just drill holes in the peptoglycan, flooding the bacteria, causing death. That's one way. Another way is they'll somehow get inside, and then they'll mess up the ribosomes and destroy the ribosomes. So antibiotics, they target certain components inside the cell of a bacteria cell or prokaryotic, and make you feel better. Now the cool thing is you're probably thinking, wait a minute, Mrs. Erickson, you just said ribosomes are found in all cells, right? And this antibiotic, maybe it targets ribosomes. Well, is it going to kill my cells?
No, but it's got
Oh, the cell ID marker? That's one way, yes.
Radiation, Eukaryotic.
The second thing, and I haven't mentioned it yet, bacteria have different ribosomes than we do. So that's another reason.
Hasn't bacteria and diseases and such become really resistant to modern?
Yes, because of mutations. And so right now we're running out of antibiotics that work. However, there's hope. We have stumbled across a super antibiotic. However, we're having a hard time culturing it in the lab. You can find it in extreme environments in nature, but then when we try to replicate it in a lab to mass reproduce it, we haven't been able to successfully do that yet.
Why do they say it's super antibiotic?
Because so far it's killed everything.
Jeez.
Yeah. So if we can just somehow get it to replicate in the lab. And I'm not saying that we should abuse antibiotics. Okay, so
Yeah,
If
saying
you are given
it's in your
antibiotics,
body
take
that-
the full dose.
it's in your body doesn't actually.
Amoxicillin is just taking a pill.
I've had my fair share.
Just drink some
Alex,
green
I'll
tea.
tell you.
Okay, so this diagram takes a look at how the staining is done. And so what they do is they'll swab your cheek or your throat, they'll smear it on a microscope slide, they'll apply the violet dye, then they'll wash it with iodine, then they'll wash it with alcohol, then they apply a second dye, the saffron, and then from there they can figure out which ones are gram positive, gram negative, and possibly what type of bacteria you have. Okay, archaea, they do have cell walls, but it's not made of a peptoglycan. We're still learning a lot about, still learning a lot about them. It's really hard to culture in lab because they are extremists. They live in extreme cold temperatures, extreme hot temperatures. They are found at the bottom of the ocean floor. They have been found in glaciers. However, common feature that you can distinguish between bacteria is they have different lipids. And so their lipids include saturated hydrocarbons that are covalently attached to glycerol at both ends. So that may not mean a lot to you right now, but on the next slide, I'll show you this monolayer that they're discussing. And this ability right here, this difference allows it to live in hotter or colder environments. However, if their environment does change rapidly, then they can't adapt and they die. So if you find something that's in an extremely hot environment and subjected to cold, it will die. So here's what a bacterial lipid looks like. Here's the phosphate head. It's charged. You got the hydrophobic tails, and you can see that it's a bilayer. There are two, right? They're not linked here. With the archaea, you can see that this one is completely linked. So they call this a monolayer. They are covalently linked together. But they do have the ability to also be bilayer, depending on which species you're looking at. So if you were to compare bacteria to archaea, so these are what the old terms, U means true bacteria, and this is supposed to be the archaea bacteria, because at one point they thought that maybe it was bacteria. These guys, cell walls, peptoglycan, cannot survive in extreme environments. Standard energy, well, don't worry about that, just scribble that out. Okay. Archaea live in extreme environments, cell walls made of different matter. And then how they are similar, no nucleus, no organelles, except for the ribosomes. Single cell, prokaryotic, it means no nucleus, and evolved from a common ancestor. Some similarities and differences. Yes!
I go to the bathroom.
Oh, I got so excited. I thought you had a question. Yes, you may. I was like, she's actually going to ask a question. Okay, sorry. All right, DNA and proteins, they're closely related to eukaryotics and bacteria. So now with technology, we've been able to sequence the genome, and we have discovered that archaea, yeah, they're similar to bacteria, but they're actually more closely related to eukarya. I love this table. It's comparing eukarya to bacteria to archaea. Talks about cell types. That's pretty important. We haven't discussed histones, so just ignore that, or introns. But the ribosomes, this is where that difference comes into play. Our ribosomes are different from bacteria as well as archaea. Cell wall composition, not always present if you're looking at animals. In plants, it's cellulose, and fungi, it's chitin. But in bacteria, it's peptoglycan, and in archaea, it's protein, but it's not peptoglycan. Okay, so prokaryotes and how they move. There are a couple different ways. One is a rotating flagella. Long thread-like structure protrudes from the surface. It's used in locomotion. It can either spin like a propeller or undulate. It can have one or many flagella. So you can see here, this is one. Here is many. They can swim up to 70 cell lengths. That's pretty fast. Okay. A second by rotating flagella like screws.
It's like a millionth of a mile an hour. If you upscale it, I bet it'd be pretty fast.
Probably. I don't even want to fathom. Okay, eukaryotic cells. So they're a little bit more complex. Some differences. Endomembrane system, which will be part 2 of chapter 4. They have organelles. Plants have a central vacuole. Now, maybe in biology they said plants have vacuoles. Was that the case, or did you always call it the central vacuole?
I remember.
Plants, it's the central vacuole.
Okay.
No.
Animal cells do have vacuoles.
Yeah.
Okay.
Yeah.
Okay, good. Because the biology book that we have states that it doesn't say animals have vacuoles. Like, that's a difference between plants and animals. Plants have vacuoles. No, that's false. Animals also have vacuoles, but it's not as big. Okay. So central vacuole in plants can store pigments, proteins, as well as waste materials. Like I said, both can have residues, smaller sacs that store and transport materials. So we'll talk about vesicles that move within the cell and how it can move things out or take things in. So we'll talk about what's the word I'm looking for? Nope. Endocytosis, exocytosis. Thank you. We'll talk about the nucleus. I'll show you a crazy video of how the nucleus moves things in and out of it. It's kind of scary. And then all eukaryotic cells are supported by a cytoskeleton. And so we'll look at the components of a cytoskeleton, and there are 3 of them. Offhand, actin, myotubulin, and intermediate filaments. So here's the endomembrane system, a visual summary of it. It's basically the ER, the Golgi apparatus, and the vesicles that kind of flow between them. And so what happens is that maybe an mRNA leaves the nucleus, latches onto a ribosome, a protein is made, that protein needs to get somewhere, maybe inside the cell, or it needs to leave the cell, and so it gets packaged into a vesicle, it leaves the ER, it travels to the Golgi apparatus, this is the vesicle, it morphs within the Golgi apparatus, forms another vesicle, and just kind of keeps traveling until it reaches the end, where it gets repackaged several times, and then will finally leave the cell in the process of exocytosis. And then on this side, we have endocytosis, where things are brought in, enclosed in a vesicle. Maybe the cell doesn't need it anymore, it might latch onto a lysosome and destroy it, break it down, I don't know. So we'll talk about the different fates of a vesicle inside a cell too. So. But first up, the nucleus. It's the information center. I'm sure you were told it's the brain of the cell. Okay, well, it just holds genetic information. It's the largest, most easiest organelle to see when you look at your cheek cells. We'll do that, I think, with mitosis. So we'll spend a couple days with microscopes. We'll look at mitosis of an onion root cell. Did you guys do that in biology? Okay, lucky you. Yay, no double them up. We'll also swab your cheek, and we'll dye it with bromophile blue. And the only thing you'll see is the outline of the cell and the nucleus. And then if you had breakfast, you might see some food particles there too. So that's always interesting. Robert Brown, 1831, described the nucleus as a kernel or a nut. Holds genetic information, leads to the making of the proteins. The nucleus is kind of broken down into different layers. So we're going to talk about the nucleolus, a dark stained zone where intensive synthesis of ribosomal RNA is taking place. So that's in the center of the nucleus. So this is how mRNA is made. And then it leaves. Okay, the outside layer of the nucleus is the nuclear envelope. It is a double layer, as you can see. So it's bounded by 2 phospholipid bilayers. The outer layer is continuous with the endoplasmic reticulum. This is really cool. This is like new stuff within the past 15 years. When a cell undergoes division, the ER literally kind of like reabsorbs by like the nucleus, and everything just kind of like breaks down, and then it reforms. So it's just so cool how the ER kind of disappears along with the nucleus when it's time to divide. Consists of nuclear pores, openings for things to get in and out. It's very complex. It's got a nuclear face, a central ring. It's crazy. I'll show you a video that breaks down the anatomy of this structure. But it allows small molecules to move in and out freely between the nucleoplasm and the cytoplasm. Passage is restricted, however, by 2 kinds of molecules. Proteins moving into the nucleus to be incorporated and to undergo nuclear activities. But then when the mRNA is made inside the nucleus, it also has to leave to get to the cytoplasm to hook up with the ribosome to make proteins. Here's the structure that I'm talking about. So we're looking at scanning. Scan an electron microscope. This is the outside surface. Electrons bouncing off, and you see things like this. And then this is kind of the transition, kind of looking through, and they're found all over the outside of the cell, outside of the membrane of the nucleus. Now, if you were to take a look at the cartoon and slice it in half, this is from the side view. So here we have our lipid bilayer for the nucleus, and then we have these really weird tentacle-like structures, and they do move like that. And then over here we have like a basket shape. So this diagram does a little bit more better surface, or better diagram. Tentacles, basket shape. These tentacles, what they do is they move, and then when things get close enough, the tentacles literally grab it, and then they just kind of like shove it through the hole, through the basket. So it's like playing basketball with itself, or dunking, I don't know. Yeah. So these fibers are intermediate filaments called nuclear lamina, gives the nucleus its shape, involved in deconstruction and reconstruction during cell division. So here's that trippy video. Enjoy. I don't think there's that much sound, actually. I think it's just like techno music or something. Maybe not. I don't know. Okay. Oh, it's trippy. Oh, no sound.
Can I get something for the announcements, please? There's a lunch menu change for tomorrow and Wednesday, there's a swap. Tuesday will be corn dogs, baked beans, mini donuts, and fruit. Wednesday will be crispy chicken wrap, Mexican rice, refried beans, and fruit. Also, if you borrow samples from Mr. Field's room, please return them ASAP. Thank you.
Now you're the protein being thrown in. I love the title of that. Into the Nucleus. Okay. Well, I guess that's where I'll pick it up tomorrow. So tomorrow I'm not going to be here. I will just videotape the lecture. So bring earbuds, because my laptop will not be here.
We could airplane to the board somewhere around
If you know how to do that. Props to you.
That'd be
Mrs. Breach
ideal.
is your sub.
Oh,
Oh,
wow. We'll bring earplugs, don't worry. You can bring earbuds.
Earplugs, I think is what we're saying.
I mean, I'll probably bring earplugs.
Which means I can't swear. Dang it.
Oh, Miss Erickson, that's what.
What?
You got Cameron and Ben.
Yeah. Why?
That'll be a good stuff before.
Oh yeah, that's what we
Oh, they just clash.
What?
Bad thing that happens. He usually ends up losing.